Good morning, good afternoon, and good evening. Welcome to my channel. My good name is Delvin Wenani. I'm a biology and mathematics teacher. I am a seasoned examiner. I mark biology, and therefore today I want to take this opportunity to welcome you to my channel. Please remember to subscribe, remember to follow, remember to comment, and remember to share. So today I've prepared paper 2, KCSE 2024, marking scheme section A. So follow me to the end. Welcome to the class. Now, in 2024 paper 2, the first question was reading, the following word equation represents a biological process that occurs in green plants. So let us look at this process. Disconnected. Carbon 4 oxide plus water, and we get glucose plus product named E. Carbon 4 oxide plus water, and we get glucose plus product E. Question A. Name the cell organelle where the illustrated process takes place in green plants. I take it again. Question A. Name the cell organelle where the illustrated process takes place in green plants. Now, before we name the cell organelle, first of all, we need to understand which process is this. Now, what we have here, we have carbon 4 oxide plus water, and we are getting glucose plus product named E. When we look at this process, it is a photosynthesis process where we are getting carbon 4 oxide being reacted with water, and actually there's presence of light there, and we get glucose plus product named E. So let us now look at where it takes place in a cell. So in this case, it takes place in a chloroplast. But we can also accept chloroplast. That is a plural of chloroplast. Question number B, Roman 1. Give the likely identity of product E. Now we have carbon 4 oxide plus water. Then we are getting glucose plus a product named E. Definitely, because it's photosynthesis, then the product named E is oxygen gas. It can be oxygen gas, or we accept the symbol for oxygen. Question number B, Roman 2, suggest ways in which the rate of product E can be increased. Now, product E is oxygen. How do we increase oxygen? Now, in this case, 1, optimum temperature will actually increase the product named E, which is oxygen. Remember, this is an enzyme control process, and all enzyme control processes, they depend highly on temperature. Optimum temperature works so well for the enzyme control activities and processes. Number 2, optimum light intensity. Now, light is required in the process of photosynthesis. Now, in this case, once we increase light to optimum, not too much, not too little, then we shall be able to increase the production of oxygen. Number 3, increasing concentration of carbon 4 oxide, or what we call the CO2. Now, when you increase the concentration of carbon 4 oxide, you also increase the production of oxygen. Why? Because carbon 4 oxide is a raw product of that process, which is photosynthesis. Increasing water availability is another requirement. Water in the process of photosynthesis is one of the raw products that is used in the first phase of photosynthesis. Now, photosynthesis takes place in 2 phases. The first phase, which we call the light-dependent stage, or sometimes we call it photolysis, or we call it light-dependent stage. This is where light is used to break water molecule. Remember, water is a molecule which has H2 and O. So light breaks the water molecule into hydrogen atoms and oxygen gas. It splits them. And therefore, the oxygen, that is where the oxygen comes from. That's where the oxygen originates. So when we increase the water, definitely there will be more splitting of the water molecules, and therefore we shall have more of the production of oxygen gas. Number C, suggest one possible source of carbon dioxide if the above process was artificially conducted in a school laboratory. Now we're saying we want to take this process, and this process is photosynthesis, but we want to take it to lab. Naturally, it occurs in plants, but we want to take it to the lab. So what are we going to, or where are we going to get carbon dioxide so that it can help us in the process? Now, in this case, because we are now artificially inducing it, we are artificially producing it, then we require to have sodium hydrogen carbonate or sodium bicarbonate in this case, or we can still have dry ice. Those ones, they can give us the carbon dioxide. They can supply the carbon dioxide for this process to take place in a laboratory, especially in a school laboratory. Now, explain how the illustrated process is significant in nature. Now, we're talking about photosynthesis. How is it significant? Now, the word significant in this case means how important is it? Now, number one, it's used for air purifications. It removes and reduces carbon dioxide. Carbon dioxide is toxic to animals, and therefore, when it accumulates, and remember, it's a byproduct of respiration. So when animals respire, and animals respire highly when they make energy, or when their bodies make energy through the process of respiration. So they respire. They produce a lot of carbon dioxide as a byproduct. That carbon dioxide is toxic to the cells of the same living organism, and therefore, when it is breathed out, it comes out and it accumulates into the air. It may end up killing, or in the other term we say, it may suffocate the same animals that are producing it. And therefore, this process of photosynthesis will utilize that carbon dioxide and therefore purify the air. Number two, increasing the concentration of oxygen in the atmosphere. As it is purifying and reducing the carbon dioxide, remember it is supplying what? Oxygen in the atmosphere. So it increases the concentration of oxygen in the atmosphere, which is used by living organisms like animals for respiration. And also it helps in the production of glucose, which is that glucose. And the glucose, which is produced by plants, mostly is converted to starch, and therefore living organisms, we get food in form of starch from those living organisms, from those plants. Now, in an experiment, the number of red blood cells, this is question number 2, in an experiment, the number of red blood cells present in 3 samples of blood labeled F, G, and H was estimated. Different concentration of salt concentration were then separately added to the samples. After an hour, the number of red blood cells present in each sample was counted and recorded as shown in the table below. Now, blood sample F was put in salt concentration of 1%, and the number of red blood cells after an hour remained the same. Now for G, was put in 0.6% salt solution, and after an hour, the number of red blood cells were fewer. Blood sample H was placed in 0.4% salt concentration, and after an hour, there was no cell that was observed. Now, let us look at the question. Now, the first question, name the physiological process being investigated in the experiment. Now, we have samples of blood being taken and mixed with concentrations of salt. Now, when you look at that, definitely there's issue of movement of water molecules because of the increase in concentrations from one cells to the other. So that is osmosis. Then account for the results made in blood sample F. Now, accounting means we have to look at the results of sample F. Now, let us look at that. The sole solution was isotonic to the cell cytoplasm. When I say cells, when I say it was isotonic, I mean it has the same concentration, and hence there was no movement of water. If the concentrations are the same, then there is no movement of water molecules in and out of the cell, and therefore the number remained the same. The number remained the same. Also, I wanted to look at it, and you'll discover that the sole solution was hypertonic to the cell cytoplasm. Question number 3. A genetic investigation in a certain population established that in human beings, premature baldness is controlled by a dominant gene, capital N, which is located on the Y chromosome. Take it again. In a genetic investigation, in a certain population established that in human beings, premature baldness is controlled by a dominant gene, capital N, located on the Y chromosome. Question number A. Work out the phenotypic ratio of the offspring of a couple comprising of a normal mother and a father with premature baldness. Question number A. Work out the phenotypic ratio of the offspring of a couple comprising of a normal mother and a father with premature baldness. Now, I want you to look at this, and you discover that the following. Now, before we work it out, I want us to analyze some things here. Number one, you will discover that in humans, sex is determined by the presence of a sex chromosome. Now, females have two, that is chromosome X and X, while males have one X and one Y chromosome. And therefore, since the gene for premature baldness is located on the Y chromosome, it can only be passed from father to son. So daughters will not inherit the Y chromosome and therefore cannot exhibit the trait. And I'm very clear on that. It can only be passed to male because this one is linked to Y chromosome. And for an individual to have Y chromosome must be a male. So if this, for example, baldness is linked to Y, so we don't expect females to have, or the daughters to have baldness, especially premature baldness, because that gene is only located on the Y chromosome. Now, after doing the crosses, this is what you will observe. This is how the results look like. That is the results for the crosses. You can pause the video and also do your own cross. You can also pause the video and look at that result. Just analyze from what we have. What we have are called parental phenotypes. We also have parental genotypes. Then we have our gametes. And then we have those crosses you can see. Then the crosses. And then we can have our offsprings. Now you see our offsprings down there. Look at it very carefully. Then I also want you to note where the markings are. The X must be there. The gametes, all the gametes must be circled. And they must be circled separately. And then the circles must be complete. If you are using a genetic cross, if it's not a Punnett square, then make sure that the crosses are completely moving from one point to another. They are not hanging. And then for the offsprings, make sure that you give us their phenotypes. Give us their phenotypes also at the end there. So that is it. You can pause and continue. Next question. What is the probability that this couple what is the probability that this couple will have a daughter with premature baldness? I've just stated that premature baldness is controlled by the Y chromosome. And since Y chromosome is only in male, there's no probability of having a daughter with premature baldness. And therefore, in this case, the probability is 0%, completely 0%. Name 2 sex-linked traits in human beings associated with X chromosomes. Now, that one was sex-linked with the Y chromosome. Now, we want to name any 2 sex-linked traits in human which are now associated with X chromosomes. And these are, number 1, hemophilia. Now, you can write hemo with HE or hymo with HAE. They are accepted. And then we have color blindness. Color blindness. We also have muscle dystrophy. Muscle dystrophy. Next question. During an ecological study in a lake ecosystem, a group of students made the following observation on the feeding relationship among some organisms. During an ecological study in a lake ecosystem, a group of students made the following observation on feeding relationships among some organisms. And these are, number 1, lobsters feed on algae. Small fish feed on lobsters, worms, and caterpillars. Caterpillars feed on algae. Worms feed on caterpillars. Large fish feed on small fish. Penguins feed on small fish, lobsters, and worms. Question. From the student's observation, construct food web for the ecosystem. From the student's observation, construct food web for the ecosystem. And this is what we have. I want you to pause the video and look at it. That is the response that we have. That is the response. And you look at the arrows. Just look at those arrows. The arrows are pointing to the eater, just as illustrated in the observation. So look at that. You can pause the video and look at it carefully. Next question. From the food web constructed in 4A above, extract a food chain with the penguin as the secondary consumers. From the food web constructed in 4A above, extract a food chain with the penguin as the second consumer. And in this case, we shall have algae being fed to lobsters, and lobsters being fed on by penguins. Algae being fed on by lobsters, and lobsters being fed on by penguins. Explain how spillage of oil from water vessels is likely to affect this lake ecosystem. Explain how spillage of oil from water vessels is likely to affect this lake ecosystem. And in this case, it will prevent exit of carbon dioxide, and this will cause suffocation and death of living organisms. Also, it will prevent entry or diffusion of air, that's in and out. So it will limit, or it will prevent and reduce photosynthesis. So very few plants will photosynthesize in that case. Also, that same oil will prevent entry of air, and air has oxygen in it, and this will cause suffocation and death of what? Organisms. Also, it prevent and perhaps reduce penetration of light. Now, if light is limited, this will prevent photosynthesis. Remember, even plants which are living underwater, submerged plants, they photosynthesize because they have photosynthetic cells which are very sensitive to light. It also clogs respiratory surfaces, limiting and also causing suffocation or death. So it will limit gas production and at the same time cause suffocation and death. Now, the following diagram represents part of a cell structure as seen under an electron microscope. The following diagram represents part of a cell structure as seen under an electron microscope. Look at that structure. And when you look at that structure, definitely this one is the cell membrane. But you can pause the video, just look at the structure again. look at the structure before we go to the questions. It has pause there, and then those are the structures we want to go to the question to look at. Now, the first question, identify the structure represented. I've already given you the hint, and definitely the answer is a what? Cell membrane. You can call it a plasmalemma. You can call it a plasma membrane. State 2 function of the structure. What is the structure of a cell membrane? Enclosing cell contents, protection of inner cell, cushioning organelles, protecting them, controls entry and exit of materials. It also regulates material in and out of the cell. Now, suggest 2 ways in which the functioning of the structure can be impaired. Suggest 2 ways in which the functioning of the structure can be what? Impaired. Now, this is the structure we are calling the cell membrane. How can it be impaired? Impaired means how can it not perform well, or what will cause it not to perform well. Now, in this case, number 1, in the extremes of pH, when the pH is either too acidic or too basic, okay, it will affect it. Also, in extremes to the temperatures, if the temperatures are too low or too high. This is because this cell structure is proteinous, is protein in nature. So being protein in nature, it means it can be affected by extreme and low temperatures. Name 2 structures found in plant cells but not in animal cells. Name 2 structures found in plant cells but not in animal cells. And these are 1, chloroplasts, or what you call the plastines. 2, subvacuole. We also have the cell wall, and we have atonoplasts. Question number C. What is the purpose of staining cells before observing them under a light microscope? Why do we stain cells before observing them under a light microscope? And the answer is 1, for clarity during observations, making structures more distinct, more clear, and more visible. Otherwise, thank you very much. That brings us to the end of section A. I'll be back with section B. I'll be looking at the graph. I'll be taking you through the graph of section B. That is question number 6. And I'll also be taking you through the 2 essays. That's question number 7 and question number 8. So, ladies and gentlemen, I want to appreciate you once more. And please remember to subscribe. Remember to follow. Remember to share and comment. Otherwise, be blessed. Let us meet in section B. Thank you. God bless you.